The coronavirus nationwide lockdown has left the streets nearly deserted. People have been ordered to stay home. Get back to what you used to do. We must set up several layers of barriers against the virus, but each barrier is not 100% effective. So to be fully protected, all barriers have to be in place. One of the key barriers is proper hygiene. Another important barrier is setting a distance between you and others who carry the virus. The other way around is also true. Protect the others from your germs. The government has also taken measures to protect the public through testing and contact tracing, quarantine and isolation, and finally, the last and most important barrier, vaccination. PGC Mindanao has been actively exploring ways to help the local efforts against COVID-19. At the end of the day, we just want to get back on our feet. Vaccination is the final barrier that completes your protection against COVID-19. So why settle for less? Be vaccinated.
Hi, good day. I'm Michelle Ann Osiones, one of the project clerk of PGC Mindanao.
Hello, I'm Marian Loayon. I'm the project assistant of PGC Mindanao.
I'm Eddie Florianong, the Project Development Officer of PGC Mindanao.
Hello, I'm Jocelyn Aurelio, Clerk of PGC Mindanao.
Bayang magiliw, perlas ng sinanganan, alab ng puso, sa litin mo'y buhay. Lupang hinirang, tuyat ka ng magiting, sa manlulupi, di ka pasisihin. Sa dagat at bundok, sa simoy at sa langit mong bagaw, mahina kang tula at awit sa paglayang minamahal. Ang kislap ng mata at mo'y tagumpay na naglilinig, ang bituin at araw niya kailan pa may maglililim. Lupa ng araw ng malakit pagsitaboy langit sa piling mo. Aming bigaya ng may mga ang mamatay ng dahil sa'yo.
Madayaw. Good day, everyone. On behalf of PGC Mindanao, welcome to the fourth and last installment of our 2022 Bioinformatics Lecture Series. This event was brought to you by the Bioinformatics Division of PGC Mindanao. I am Jerouen Paul Lumabao, JP for short, and I will be your host for today's session. I hope everyone is doing well and ready for our event today. Before anything else, let me take you to a quick review for our previous lecture sessions. Our first lecture had Dr. Victor Lopez, who talked about the intertwined paths of bioethics, genomics, and bioinformatics. In our second lecture, we had Mr. Kevin Libuit, who discussed virtual resources for global public health bioinformatics workforce development. Down to our third lecture, Mr. Frank Ambrosio tackled the evolution of public health pathogen genomics. Today, we will be featuring a medical microbiologist and a medical doctor based in Canada, who is also the CEO of a bioinformatics company named Bugseq, a leading company in the adoption of sequencing for infectious disease diagnostics and public health in the world. Before we start, I would like to remind our participants that if you have any questions while the talk is going on, you may place them in the Q&A box of Zoom, after which we will have a question and answer portion after the talk where we will try to read these questions for the speaker to respond in the allotted time. So warm up your bowl of poutine and let's get ready to listen. To start off our last lecture, let me introduce to you the moderator for this session. He is currently the Junior Research Analyst assigned to the Bioinformatics Division at PGC Mindanao, where he is in charge of research, client, and training services related to bioinformatics. He received his bachelor's and master's of science degrees at the University of the Philippines, Diliman, in the fields of molecular biology and biotechnology. His research interests at present include bioinformatics, omics, industrial biotechnology, and molecular medicine. Everyone, please help me welcome our moderator, Mr. Paul Lorenzo Gaite. Take it away, Sir Paul.
All right. Thank you, JP, for that kind introduction. Good morning, afternoon, and evening to everyone, wherever you may be. And thank you to PGC Mindanao for having me as your moderator for today's webinar. It's definitely a pleasure and an honor. Our speaker for today is definitely no stranger to me. In fact, Dr. Sam Chorlton is our collaborator in the recently concluded Bill and Melinda Gates Foundation-funded project through the Public Health Alliance for Genomic Epidemiology, or PHAG, as a grantee, and with PGC Mindanao as one of its sub-grantees. Sam is the Chief Executive Officer or CEO of Bugseq, a Canada-based bioinformatics company leading the adoption of sequencing for infectious disease diagnostics and public health. Sam is also a medical doctor with research expertise in bioinformatics. He is also a board-certified medical microbiologist in the US and Canada. So without further ado, everyone, please help me give our warmest Filipino welcome to Dr. Sam Chorlton. The virtual floor is now yours, Sam.
Thanks for the introduction, Paul. I will share my screen. Awesome. Okay. Doesn't want to share. Try this one other way. One sec. Paul, can you confirm that you can see that?
Yes, we can see your slides, Sam.
Perfect. Okay, so thanks everyone for joining me, and it's a real pleasure to be invited to this talk where I'll be talking about applications in bioinformatics at Bugseq. And Paul already gave a very elaborate intro on some of my background. It seems like Zoom is not wanting to collaborate. Let's try. Just give me 1 sec to see if we can make this bigger for you. Is that okay for you? Hopefully that's okay.
Yeah, that's fine, Sam.
Okay. So I'd like to just acknowledge the PGC Mindanao team. It's been an absolute pleasure to work with them, as well for the invite. So as my Bugseq team, I wouldn't have been able to present all of the work that we're going to talk about without that team. That includes a team of software engineers, bioinformaticians, microbiologists, and admin. And then our users at Bugseq, so that's the clinical and public health labs. And then finally, 𝗕𝗿𝗄 for funding the collaboration with PGC Mindanao, where there's a lot of learning to be had at Bugseq, and I think hopefully across all of the partners on this project. And so as a background, we know that analysis costs time, accuracy, and health. And specifically, I'm talking about bioinformatics and bioinformatics within clinical and public health microbiology. So we know that there's many open source tools, and it's hard to get them working. So you have to know command line knowledge, you have to know how to program, you have to figure out all the flags. There's lots of intricacies to it. And not only that, but many people will be doing the same analysis every time. And so, you know, a lab in Philippines may be doing the same SARS-CoV-2 analysis as a lab in Canada or the US. And so then everybody's reinventing the wheel. It can be hard to get compute infrastructure. So some of the analyses that Bugseq run requires over 1 terabyte of computer RAM. And so getting that kind of infrastructure on site can be complicated, or hundreds of CPUs, or the storage for all of the genomic data. We know that analysis can take a long time. That's similarly related. And there's multiple factors there. One is getting pipelines up and working. One of them is getting the data pushed through the pipeline. So is that automated? Do you have a person? And the third is, do you have, again, the infrastructure to scale that analysis to run it really rapidly? And similarly, labs need quick results. When you're in clinical microbiology, we know that mortality from sepsis increases for every hour that the patient isn't on appropriate antibiotics. And so it's vital to get accurate and timely results when you're doing tests for clinical microbiology. Same for public health, where we know infections are spreading in the community, and genomic-based epidemiology has the potential to inform infection prevention and control measures, and so rapid results are necessary. And finally, debugging is painful. So if you've gotten these command line tools working, or if you string them into a pipeline and it doesn't work, figuring out what's going on can be challenging. So here's our mantra at Bugseq, where we want to enable sequencing to enhance all microbiology tests. And so the lab sequences the sample. We don't do any sequencing ourselves. All of our partners, customers, and collaborators do that. And so one example is a bacterial isolate, but I'll talk about some other examples later. They'll upload that data to Bugseq. So we have a cloud platform. We'll take that data, perform quality control. We'll detect all of the pathogens in the sample. So that may be a single pathogen if it's a bacterial isolate. That may be multiple pathogens if it's a metagenomic sample. We'll predict antimicrobial resistance for each pathogen in the sample, even in the presence of thousands of background microbes. So we know that many body sites have normal microbioflora. So for example, the stool or the nasopharynx or whatever it is, the lungs. And so we can subtract all of that normal flora and really pick out those pathogens, which are important from a clinical and public health perspective. And then we can characterize them even further to track their spread in the community. So if it's SARS-CoV-2, we can do variant of concern and lineage typing. If it's influenza, we can do lineage, strain typing. It really doesn't matter, and as well as bacteria, to see the spread of bacteria in hospitals. And so really this enables labs to meet their mandate of serving clinicians and epidemiologists and improving the health of patients and the public. So I'll show you a quick demo. Let's try sharing a different window. So here's how data submission looks to Bugseq. And it's as simple as uploading files to our platform. You can drag and drop, you can select. We also have integration with some cloud providers like BaseSpace for Illumina. You can name your analysis. You can say, we support all major sequencing platforms, whether that's Nanopore or Illumina. So you can say, I want to do Nanopore, as well as what platform you sequenced on. There's a database selection. You tell us what type of sample you type. So let's say I did a upper respiratory sample, and I did RNA sequencing, and then you just hit submit, and then you're good to go. And so within 2 hours, we'll provide back results, often within 1 hour, of exactly what I mentioned before, what's in the sample and characterization. So here's an example where a user submitted 3 bacterial isolates. And so you can immediately see that they were all 𝗕𝗶𝗃𝗃 pneumoniae. And we performed automated assembly. And so we could assemble the complete genome, which is 5.8 megabase pairs for 𝗕𝗶𝗃 pneumoniae. And this is the reads passing quality control filter. So it's all quality controlled, processed completely automatically without any user intervention. There's things like plasmid detection, because we know antimicrobial resistance spreads on plasmids. There's detection of genotypic markers of antimicrobial resistance, as well as a predicted phenotype based on that genotype. So at the drug level, for each sample, what drugs are going to be resistant versus, or predicted to be resistant based on a genotypic marker versus predicted to be susceptible based on the absence of a genotypic predictor. And so this is all reported in accordance with clinical guidelines and public health guidelines, where we know certain drugs are appropriate for certain organisms. I also talked about metagenomic classification. And so this is from an assembly-based approach where we assemble the reads into longer, higher quality contigs and then classify that. But we also do a read level classification. And so we can immediately see for all 3 samples that we have 𝗕𝗶𝗃𝗃 pneumoniae here for all 3 of them, and that they're pure samples. The gray other is sequences deriving from plasmids in these samples, so they're pure. And then there's quality control metrics, including number of contigs, read quality control, sequence quality, etc. And then taking it a step further, you can actually dive into each of your samples. And so here's an example of one of those 𝗕𝗶𝗃𝗃 pneumoniae samples, and we can do a deep dive on all of the plasmids in that sample to see, are those plasmids circularized, how long are they, the number of contigs, as well as their host range, as well as when we look at antimicrobial resistance, we can actually tie the genotype to a phenotype. So for example, for ampicillin, we can say it's resistant because there's a CTXM in there, there's an NDM5, an oxonine, etc. So we really tie that genotype to predicted clinical resistance. Scrolling down, there's more again on, strain typing, whether it's 𝗕𝗶𝗃𝗃 pneumoniae and hypervirulence typing, as well as quality control data. So I'm going to stop that there. And that's a quick overview of the Bugseq analysis. I'll go back to slides here. That should be it. And so some common applications. Okay, hopefully that's okay. Some common applications of Bugseq are antimicrobial resistance prediction, whether that's for very resistant organisms like carbapenemase producing organisms, which are resistant to one of the last line antibiotics or the last class, which are carbapenem antibiotics. Tuberculosis is another popular one, because traditionally it takes a couple weeks to a month to grow Mycobacterium tuberculosis, and then another couple weeks up to a month to do antimicrobial susceptibility testing. And so you can imagine for the patient is on empiric therapy for a month and a half or potentially longer before they actually know that they have tuberculosis and that this is the appropriate therapy for them. And so by doing sequencing, there's the potential to rapidly speed up antimicrobial resistance detection. Other popular analyses that Bugseq serves are hospital-acquired infections. So we work with hospitals as well as state labs across North America and the world to track the spread of bacteria between patients within the hospital. And again, that can be organisms like carbapenemase-producing organisms, as well as more common ones like methicillin-resistant Staphylococcus aureus or vancomycin-resistant enterococci. Organism identification and metagenomics. So if our users sequence something and they either don't know what it is because traditional methods could not identify it, like their MALDI-TOF or their VITEC or biochemical testing, then we can do that via whole genome sequencing. And metagenomics, so the identification of pathogens in samples with many organisms. And then some of the other ones are amplicon sequencing, so things like 16S sequencing for bacterial identification direct from specimen, and viral strain typing. So one of the questions that I got from the PGC team was, I'm a medical doctor, what am I doing at Bugseq, doing bioinformatics? And so here's me growing up working, and I started on computers at a young age. And I'll try and, I see some reactions there. Oh, I think those reactions actually to freeze Zoom. Oh, okay, there we go. So I started with a Bachelor of Health Science, very generic, but during that I started with bioinformatics and doing research on kind of extra time in bioinformatics because I had a passion for the interface of biology with computers. Then, and I'll get into this more in a second, then did my Doctor of Medicine, specialized in medical microbiology, as I had, again, an interest in the systems level impact that can be had within healthcare. And it was also an interesting area at the interface between bioinformatics and microbiology, which I'll get into. And that's really how Bugseq was born, was to serve these clinical and public health microbiology labs and specifically enable them to perform sequencing, as bioinformatics was one of the last remaining barriers that we saw. So when I was doing my residency, what I saw was one hospital started doing sequencing, then they gave me their data to analyze because they struggled with that process. Then another hospital did that, and then third hospital, and then it was no longer scalable. And so that's when we built Bugseq to enable them to submit the data and automate that entire process, and it quickly grew from there. So in terms of my first bioinformatics experience, I was actually building models of gene regulatory networks in a French lab. And so I had no experience at this point. So I kind of just sent out my resume everywhere, but I had a bit of programming experience from high school. And so I just dove straight into it. Then when I came back from this French lab, I was fortunate to then have some experience on my CV. So I was like, sent my CV to lots of labs and got a position in a lab at McMaster University doing gene expression analysis of tumors. Again, not microbiology related, but there are some common themes. And so at that point, I was still on microarrays for looking at gene expression, but the technology was in the very early days of shifting to RNA sequencing for gene expression analysis. So that's where I got my first introduction to sequencing. And so then the other question that I got was more along, how did I get to Bugseq? And so surrounding yourself with smart people. So this is actually my brother, Josh. And so I was fortunate that he was visiting me once, and then he had a background in tech and was really the tech brains behind Bugseq and put up our first website and minimum viable product and got our first users on board with that. And so really surrounding myself with smart people was very valuable. And so I think Paul and Shimmy and a bunch of the PGC people had asked, like, getting into bioinformatics and getting a program set up is very challenging. And I think what I've seen and what we've seen at Bugseq is that it's very hard to move from a career in biology to bioinformatics, and also a career in computer science into bioinformatics. It's this weird interface of the specialties. And so when you're trying to run a tool or get an analysis going, we see it as a spectrum where at the easiest end, and this is the market that we work with, is use an established pipeline with a graphical user interface. So you can just go to our website, you can perform a data drop. Everything is completely automated, yet tailored for your analysis. So I showed you how you can select the sequencing platform, and that will actually affect what kind of analysis we do so that it improves the results of Bugseq. Kind of the slightly harder thing is to send it to a friend, and that's because you need to communicate with the friend or the colleague about what are the data characteristics. And so every data will have unique characteristics, and the analysis needs to be tuned for that. And so if that tuning can't be automated, then you have to convey that. Designing a workflow within a graphical user interface. So that's things like 𝗕𝗿𝗄𝗕 platform, which are really great if you have some general background and you can piece together some kind of pieces of the pipe. Then there's established pipelines with command line interface. And so this might be things like the 𝗕𝗏𝗄𝗕 interface, where you need to know how to program a bit to get it up and running. And finally, the hardest things are to code a workflow yourself or build a tool, which is really the hardest one. The other thing that we've seen is brand and reputation. And so I appreciate the video intro of PGC Mindanao, and as well as these webinar series, because I think it's really, really impressive what you guys are doing. And so we've been trying to follow suit and build brand and reputation at Bugseq. And so really showing the science behind what we're building. And so we try and publish it and present at conferences as much as possible. One question from the PGC Mindanao team that was given to me earlier was how to build industry academia partnerships. And I come at this with a different lens, mainly because I was in academia before through medicine and residency, and then now in industry at Bugseq. And so the key things that I've seen is you need to either have a problem or a solution, or sometimes both. And so as an example, at the start of the pandemic, we were doing SARS-CoV-2 analysis at Bugseq because everybody was doing SARS-CoV-2 sequencing. And PGC Mindanao was looking to build infrastructure for SARS-CoV-2 sequencing and analysis. And so we could use Bugseq as a comparison to validate some of the bioinformatic methods, which was really exciting for us to work with PGC Mindanao. And then we've also developed a lot of our partnerships through conferences and networking, which has proved very fruitful. So here's a couple examples of our partnerships and how they work. So one partner of ours is the Mangus Lab at the University of British Columbia. And so Amy and Nick are pictured at the bottom. Nick is a PhD student within the Mangus Lab who we work closely with as well. And so they're really experts in metagenomics. They have a profound experience in using sequencing to identify organisms in samples, as well as using that data for epidemiological and surveillance purposes. The Mangus Lab is based at the British Columbia Center for Disease Control, and so they have a unique perspective on the epidemiology and surveillance of viruses as well as other pathogens within the jurisdiction of the provincial public health lab. And so we at Bugseq consider ourselves to be experts in bioinformatics and clinical microbiology. And so this was a unique opportunity to put together the metagenomics as well as the bioinformatics and clinical micro. And so putting this, we see this as an opportunity to change diagnostics as we see sequencing quickly taking over a lot of the diagnostics in microbiology world. And specifically, the reason we see that is going back to the beginning where there's several key challenges. Sequencing has largely been tackled. You can now buy a MinION sequencer for $1,000. Sample preparation is still a bottleneck, but it's improving as sample preparation methods get easier as well as automated. And bioinformatics is becoming automated, user-friendly, cloud scalable, and that's really the push that Bugseq is making. And together we see this entire environment where you have this easy sample prep, sequencing, and analysis to produce clinical diagnostics. And so one of the limitations specifically with clinical microbiology diagnostics is they're targeted. And so they only test for specific pathogens, which are selected before testing. And so an example is SARS-CoV-2. If you think you have COVID, you might go to a testing lab and request, or a doctor may order SARS-CoV-2 testing, but it only tests for SARS-CoV-2. And the test may additionally have included influenza and RSV. So you may be tested for a couple of viruses, but those all have to be specified in advance. But if it's a rare pathogen, so a virus that doesn't come around often, or novel, or emerging, or simply a virus that we don't test for, like we don't test for human metapneumovirus, as well as parainfluenza viruses and adenoviruses very often in the common population, then those will all be missed. And so because of this targeted approach, current diagnostics are unlikely to detect the next pandemic agent. And so we know the next pandemic is again likely to be a respiratory virus. It's again likely to be an RNA virus. And so we need diagnostics that can detect this on day 0 of when it starts spreading. And detecting it earlier is more likely to help humanity contain it, as well as develop countermeasures so that if we can't contain it, at least we can, for example, develop vaccines sooner. And so here is just a brief background on what metagenomics is and kind of how the data looks. And so all of the DNA or RNA in a sample will be extracted and sequenced. And so on the left here, you may have a community with multiple different bacteria in it. And when you extract it, you get all of their DNA mixed together in a pot. You sequence all that pot of DNA, and then on the analysis side, you can split it up and say, okay, this piece of DNA came from E. coli, this piece of DNA came from Staph aureus, etc. And so when you visualize that on the right, you can generate things like corona plots, which show the breakdown of where all of the sequencing reads came from in the sample. You can imagine that if you do a nasopharyngeal swab without any host depletion, 99% of those reads will come from human because you swab the back of somebody's throat or nasopharynx, but 1% may come from a virus. And so that's the part that we're interested in, where it's like, hey, this 1% comes from, virus X, which is the new pandemic agent. And so we started with the Mangus lab on this premise where we could use metagenomic sequencing in a widely deployable platform, which was nanopore sequencing for detection of respiratory viruses. So this was our first publication with them, which showed that we could detect SARS-CoV-2 in clinical samples. And here's the summary. So we did random sequencing of RNA in the sample on the nanopore, and it samples less than a cycle threshold value of 30 on PCR. So these are relatively high viral load samples. We had very good detection, 95% sensitivity on these samples. Above cycle threshold of 30, so these are pretty low viral load samples, the sequencing had very poor detection, so approaching 0%. Similarly, there was 100% specificity in our analysis. So in no samples did we say there was a virus when there truly wasn't a virus present. When we looked at comparing the percentage of reads in the sample that map to the virus versus the known abundance of the virus in the sample, or semi-quantitative abundance because it's not quantitative PCR, we saw that there was a pretty good linear correlation between the number of reads in a sample mapping to the virus and the cycle threshold value on PCR. And so we use this as the foundation of the work to secure a contract with the US government to further develop an assay for pandemics, as well as unknown respiratory agents. And so that's really what we continue to work on to this day with the Mangus lab, is optimizing this technology to really pave the way for the future of diagnostics and clinical microbiology. Here's a second example. And so this is with Providence Healthcare, which is a acute care hospital system based in Vancouver, Canada. And the team at Providence Healthcare are really leaders in virology. And so they have a centralized virology lab where they do most of the virology testing, or a significant part of the virology testing, for a large population. And these are collaborators pictured at the bottom, Gordon Ritchie and Chris Lowe. And what we know, one of the things that they're working on is cytomegalovirus. And so cytomegalovirus causes significant morbidity and mortality in transplant patients. So this could be everything from stem cell transplant patients to kidney transplant patients, where these patients are severely immunosuppressed. And cytomegalovirus, which is normally not that harmful to humans, it causes kind of like a flu-like illness, can be fatal in these transplant patients. And so in transplant patients, it will be treated with antivirals. The problem is that prolonged treatment and repeated treatment can cause antiviral drug resistance, where the virus evolves to replicate even in the presence of the drug. And so there needs to be a way to predict that antiviral drug resistance and detect it. And so we worked with Providence Healthcare to change the paradigm. So the current paradigm is based on Sanger sequencing, which they are sending out to an external lab. And the turnaround time on that was over a week long. And so we said, hey, we can do this in the lab, in-house, rapidly, again on a MinION sequencer or a GridION sequencer on nanopore sequencing, to produce the same or even better results. And so that's where this publication came in. So we performed next generation sequencing for cytomegalovirus antiviral resistant genotyping clinical virology lab. And so we did this on a nanopore sequencing, and we even looked for minority variants. So you know that in a population of viruses, oftentimes they'll all share the same mutations, but sometimes only a fraction of them will have the mutation. So for example, like 10% of the viral population. But those viruses can then become the dominant population if exposed to antiviral. And so you even need to detect those 10% or sometimes 1% of the viral population. So what we showed is that we had 100% sensitivity to detect known mutations. These were consensus, so seen across all the viruses in the same patient. So across 20 patients, we detected all the mutations. But in 6 of 13 samples, we also detected additional mutations conferring resistance, showing that the next generation sequencing was better than the traditional Sanger sequencing. And we cut the turnaround time by over 5 days, enabling patients to get on better, more appropriate therapy sooner and enable better outcomes. So at this point, I just want to pause. I'll say thank you, and I'll open up to any questions, comments, and thank you again.
All right. Thank you very much, Doc Sam, for that enlightening and very informative talk. So I gathered a lot of insights actually from what you just talked about earlier. And okay, so I will open the Q&A box right now and take a look at the questions that our attendees have given. So the first question here by anonymous attendee. So he or she asks, do you think bioinformaticians who develop tools or the programmers are more superior than those who have only utilized online tools, especially those who don't have programming skills? So I think this is more of a question on like differences in the background of bioinformaticians, like if they're from a non-computer science background, those who don't have a programming skills, or versus those who come from the computer science background, or those who are very proficient in programming. So do you have any thoughts on this, Sam?
Yeah, so our thoughts are that everybody brings valuable knowledge and experience to the table. So the biologist will understand the biology really well. The computer scientist will understand the programming really well. And so one thing that we see is that bringing those two interfaces together, and really that kind of intersection, is where the value is. In terms of the actual tools, unfortunately, many bioinformatics tools that are out there, like open source tools, are written for academic projects, and they may be built by one developer and then abandoned after that, or not maintained properly. So getting other tools to run properly can be very challenging in our experience. And so that's where we see the value of the computer scientist come in, or the software engineer who knows how to, for example, lint code, perform continuous integration, continuous development, keep that high quality code. But then understanding, for example, like what is primer trimming, how to identify antimicrobial resistance, all those mutations, that's where the biologist and bioinformatician come in. And so really that intersection is where we see the value of building the bioinformatics of the future. And yeah, I'm happy to chat more, like if you're interested in getting involved, because we really appreciate all the different perspectives.
Thank you for that, Sam. Another question here from another anonymous attendee from the Zoom chat. Do you currently practice medicine, or are you a full-time bioinformatician? If it's okay, may I ask why you chose to go to med school instead of taking a bioinformatics master's or PhD or something similar? He or she is asking as someone who is choosing between the two paths. So you might have any advice for this person here who's trying to choose between being a researcher versus being a medical doctor?
Yeah, that's a great question, and I can relate. So to answer the first thing is I do maintain a valid medical license, which requires a very small amount of kind of practice, almost you could say. But that is very valuable to be in terms of Bugseq's position, because a lot of what we do is basically clinical microbiology or public health microbiology. And so it's vital for Bugseq to stay up to date and really practice evidence-based analysis. In terms of how to pick, I'd really say follow, I mean, this isn't that helpful, but follow what you enjoy. There's no right path, for example. And think about how kind of your impact in the world, and that at least that's how I think about it. For me, is how can we scale our impact and really make the world a better place? And that's really what we strive for. So I don't know if medicine is the right approach for that. I don't know if bioinformatics, or potentially both, you can make a big impact. And so really following that.
All right. Thank you for that. Well, personally speaking, I'm really more inclined towards research, so that's why I'm here as a bioinformatician at Philippine Genome Center Mindanao, and I plan to actually want to do a PhD myself, since research is really my path. So I think it was like few years ago, where I realized that, yeah, I think this is really the path for me. So I chose this rather than a medical doctor path. So thank you. Thank you for that question, anonymous attendee. If I may, I have a question myself to ask Doc Sam here. So you've mentioned, Sam, previously that diagnostics are rather very specific. So, for example, you might have a SARS-CoV-2 or influenza. So, diagnostics approach is rather specific. So it really specifically looks for those for specific pathogens. And so I would like to ask, is it on the sampling or the wet lab part, wherein you sample your pathogens of interest, versus, for example, in the bioinformatics side, wherein you have to detect, I would imagine it's a metagenomic sample. So you take the DNA, then you map it to known databases or known sequences of the pathogens, so that you'll be able to detect those pathogens. So it's rather more like wet lab versus a dry lab, on like which, in your observation, which is more likely to, because you mentioned that you wanted to detect rare pathogens. So which of these, wet lab versus dry lab, is would like you would get more, like you would detect more of those rare pathogens? So is it the wet lab or the dry lab, or is it both? So do you have any insights on that?
Yeah, so the current reason why current diagnostics are limited is because they're mainly PCR based. And the PCR is a targeted PCR looking for, so that the primers are for specific pathogens. And so it's purely based on the wet lab approach taken. And that's because PCR is well established. It's been in use since the 90s. It's cost effective. You can test samples for a very cheap amount. It's highly sensitive and specific, so it has really good test performance characteristics. The one drawback is you can't detect everything. You only detect the thing you're looking for. And so that's kind of where the sequencing comes in, and where the future might be, where it's improving sequencing so that's as sensitive as PCR, as cheap as PCR, but can detect everything. And that's kind of the future that we're working towards.
All right. Okay. So thank you for that answer. So okay. So I'll take another question here on the Q&A box. So this question, do you have any advice for people who are starting out with bioinformatics and aspire to work in the field in future? Any thoughts?
It's a wide open field right now. There's a lot to do in bioinformatics, everything from research to applied. So just pursue your interests, I guess, get involved in the community, whether that's in academia, in research collaborations, in industry. There's so much opportunity. And if you're interested, definitely get in touch with me after. I'd be interested in chatting too.
Yes, I think we will, if it's okay, yeah, we will give out your or we'll give out your email address, or people can get in touch with us through PGC Mindanao. Then we will send that message to Sam. Yeah.
Awesome.
Yes. Maybe personally, I would like to chime in on that. Advice for people who are starting out with bioinformatics. Yeah, if you're like from a biology background, like myself, so I started out actually, my story was that I was choosing between the wet lab or versus the dry lab, because I tried out wet lab, but for me it wasn't really for me. So and then before, because I had my master's in marine science, major in marine biotechnology. So I got to do dry lab and wet lab as well. And I tried out for wet lab, but I didn't like it. So I dove in the dry lab, and the bioinformatics side of it. So I explored. I also tried did coding. Actually, I joined an internship before. It was like a bioinformatics focused internship, and that's where I really learned a lot, and I got to really practice my bash or command line skills. And also we had also a project to do for as a requirement for finishing the internship, and that's where I learned a lot, and I got really practice my command line skills, coding skills, and then I was already a bioinformatician here at PGC Mindanao, and so I really got to practice those skills, and that's where it got me here. So now I have some I have to admit I still have a lot to learn about bioinformatics, because really bioinformatics is a really wide field, and maybe a few years is not enough really to like be a full-fledged bioinformatician. Yeah, I still have a lot to learn. So yeah, so just keep it up, and yeah. So thank you for that question.
Thanks for sharing your experience, Paul. And for those who haven't worked with Paul, he's a wizard on the command line.
Thanks. So okay, so here's another question. So what does the future hold in terms of using machine learning in bioinformatics? I was wondering, what are the possible developments with the use of machine learning in bioinformatics?
Oh, there's a huge untapped area for machine learning in bioinformatics. So I'll talk about some of the ones that we're excited about at Bugseq, specifically again with our focus on applied microbiology. And so one of the biggest ones is predicting antimicrobial resistance. And what we see is that traditionally people have used kind of binary classifiers to predict resistance. So is this gene present, yes or no? Therefore, if it's present, there's resistance to the antimicrobial. If it's not present, there's no resistance. Same thing for single nucleotide polymorphisms, where if this mutation is present, then it's resistant. If it's not present, it's not. But really, that's a very simplistic model of antimicrobial resistance in bacteria. And then moving towards viruses, fungi, parasites is even more complicated sometimes. And so there's going to be this huge move towards machine learning for antimicrobial resistance in bacteria, or in microbiology. Another thing that set the stage for this is we now have the data for it. So there's now public repositories where you have both sequencing data, which is now much cheaper to accomplish and share, as well as the phenotypic susceptibility data. And so putting those two pieces together, you can build models now on really like large amounts of data. So there are certain organisms where this is particularly important. I mean, I think many people within microbiology know that Pseudomonas is a very clinically important organism. It causes a large morbidity and mortality. And it's also very, very hard to predict antimicrobial resistance of Pseudomonas because it has many mechanisms, whether that's beta-lactamases, efflux pumps, porin mutations, etc. And so machine learning models are going to be very important for accurate prediction of that problem. But there are other areas within microbiology as well. So determining if organisms are related to each other or not. Traditionally, whether it's for viruses or bacteria, when we want to assess the spread of organisms between patients or people in the community, we look at the number of mutations that are different between the two genomes in the two patients, or the number of alleles that are different if we're looking at like a core genome MLST or whole genome MLST based approach. But assigning a threshold to say, yes, like this organism spread from person A to person B, as well as determining like, is there missing people that we haven't sequenced, and maybe it spread from person A to B to C, and identifying those transmission networks, as well as the timeline for that transmission. There's going to be a lot of machine learning that's going to be needed to that problem as well. both for investigation of known outbreaks, but also surveillance. So can we take all this genomic data, monitor in real time to find the outbreaks and stop them before they grow bigger? And that's really going to be an important part of infection prevention and control. So in summary, there's going to be a huge area for machine learning, and we're very excited about it at Bugseq.
All right, thank you, Sam. Personally, I find the machine learning and artificial intelligence also an interesting topic. But I myself, from a life science or biological background, I'm not really that familiar. Even up until now, I'm still trying to learn coding and because you really need coding, programming experience or skills to be able to tap into AI and machine learning. So yeah, I really find the machine learning and artificial, and they're already there's much talk about AI and machine learning as being also being the future of bioinformatics, actually, in life sciences, since there's really a lot of potential, as Sam here mentioned earlier. So thank you for that question.
And Paul, if I could actually just build off of what you said,
Sure.
it kind of brings back a lot of thoughts,
Mm-hmm.
where it's like, we might have these models that predict that organisms are antibiotic resistant to certain drugs.
Mm-hmm.
But if it's machine learning and black box, then it's very hard for people to trust a black box if you don't know what goes on under the hood. And especially like us biologists, we're like, how did it come up with that, prediction? And so there's going to need to be a shift in mentality to how we view these models, so that they're more broadly adopted. Because currently, like in I would say a common perception is that the one gene antibiotic resistance or one SNP antibiotic resistance. But then once we get into the super high dimensional data, moving towards a simplifying understanding of that model prediction is going to be very challenging.
Wow, thank you. Thank you, Sam. Okay, so let's take another question. So this is actually from one of our very own. So it's from Dr. Alela de Cadiz. I think you've met her before, Sam. So she asks, so thank you for the interesting talk. So her question is, for epidemiological surveillance of food and waterborne diseases, especially the rare pathogens you mentioned, should we focus first on clinical samples or environmental samples? Or is it better to do all sampling at the same time, following the One Health approach?
I guess the short answer is depends on your purpose. For food and waterborne diseases, it's hard for me to comment which ones would be better. There's clearly value in a One Health approach for many areas of microbiology and disease, whether that's food and waterborne or for respiratory pathogens, etc. I don't know specifically for this one. What I can say more broadly, though, is for pandemic surveillance, we see value in both. So there's clearly going to be value in the clinical market where people are presenting to healthcare with symptoms, whether that's like critical illness or more just general respiratory symptoms, or even if it's not a respiratory pandemic, but another syndrome. And so those people will be pre-selected, and then you can figure out what they have, hopefully with better diagnostics, and identify the new agent or the rare agent sooner. But there's also value in that environmental surveillance. And so that may be kind of more distally at wastewater surveillance. We know, from SARS-CoV-2 that there's a lot of value in seeing what's circulating in our community, whether that's strains, but you can look for other pathogens in wastewater as well, or going more broadly out to other environmental sources. There are obviously concerns how far out you go. You don't want to bring back the dangerous things into your community after you go out to find them for environmental sampling. And so I think there's value across the entire spectrum. Beyond just the value of where to look, there's other things like sustainability of surveillance. And so there might be ulterior financial kind of incentives to do sequencing within a clinical setting versus an environmental setting where it may be more like a government system that's set up similar to our firefighters, where we just pay this government system to protect the community at large. And so it's the model of how it's going to work is going to change, but the overall value from all of them is definitely there.
All right. Thank you for that answer, Sam. So, okay, so let's take another question. So one asks here, will bioinformatics boom anytime soon? So what are your thoughts on this, Sam? So thank you.
I think it's booming now.
Yes, I also agree. Yes.
I think it's a great time to be a bioinformatician. And yeah, I look to Paul and the other PGC Mindanao team on their thoughts on that as well. But what we see of Bugseq is people are rapidly adopting sequencing, and not only within the infectious disease space, but it really across all spaces, whether that's other health domains, so for example, cancer, non-invasive prenatal testing, or whether that's outside of health as well for environmental purposes, agriculture, basic scientific research. Sequencing technology has rapidly advanced within the last 20 years. So the cost of sequencing has come down significantly. The output of sequencers have gone up, and the ease of sequencing has improved dramatically. And so with this uptake of sequencing, there's all the sequencing data that needs to be analyzed. And some of that will be automated with tools like Bugseq. Some of that will be completely driven by bioinformaticians and like really research-driven questions. And so it's a great time to be a bioinformatician. I think the boom is now. I don't know if it's going to, what the future holds, but I'm excited about the present.
Yeah. So taking off from what Sam has mentioned, really, yes. Well, I myself actually is very busy right now. I'm really, processing a lot of bioinformatics data. That's because due to advances in next generation sequencing. In next generation sequencing, really you're dealing with a voluminous amount of data. Like I can say on the top of my head, I'm handling like the largest assembly that I've done. That's only for a mitochondrial genome. It's already like about 400 gigabytes of data. That's just one run. And the data that I'm handling right now is ranging in that size. And so you have all these data, and there are really millions of species out there in the world. And so we have all these improving sequencing technologies right now, and you can sample any one of those millions of species, so we really have the millions to of samples possibly that you can sample and sequence. And each of those will hold a lot. You can get a lot of data, like what I said, gigabytes of data each of those samples. And it's really that's why I think that's driving the boom in bioinformatics at present. Yeah, so thank you. So another question here. So I think we have a lot of questions lately. I'm seeing here in the Q&A box. So here another question is Bugseq free and accessible to anyone? So perhaps any thoughts on that, Sam?
So get in touch with me offline. So I'd love to chat with you if you're interested in using Bugseq. We definitely work with collaborators across a different kind of models. And so my email is just sam@bugseq.com, and shoot me an email and we can set up a chat, and I'd love to learn about your project and work something out.
All right. Thank you. Thank you, Sam. Here's another question. He asks, can bioinformatics be applied in conjunction with electron microscopy? So I think you might have an insight on that, Sam, on that question.
In conjunction with electron microscopy?
Yes, that's what he asked. He or she asked.
I don't know. I'd be interested to hear from the person who asked the question. If they have a specific application in mind. I'm sure there would be ways to combine the data. So whether that's like data or image processing from electron microscopy, I'm not sure if that would fall under bioinformatics, but it's some kind of data analysis within the biology realm, all the way to if you're also sequencing the sample and you have electron microscopy imaging of the sample, then perhaps there are ways to integrate that data across multiple data sources. And so I would love to hear from the questioner if they have a specific application in mind.
Actually, we had from the first bioinformatics lecture series, we have a lecturer talking about image processing, if I believe. So his name is Dr. 𝗕𝗿𝗿𝗶𝗃 from, he's also based in the UK. So I might want to the one who asked the question to refer to that talk. I think, yeah, the recording is just available at the PGC Mindanao Facebook site. The recording, the full recording of that lecture is available. So I might enjoin the one who asked that to refer to that lecture, so you might get insights on your question. So all right. So are there any more questions? Maybe maybe two more questions. I have a question. This is not really so much on the bioinformatics side of things, but rather because like you mentioned, Sam, and actually we've been collaborating with Sam and Bugseq on this, is that their aim is to like operationalize bioinformatics especially in low to middle income countries such as the Philippines and other, maybe in African or in other Asian countries. So I would like to ask, how would you address the, like, yeah, we've been talking about this. On our side, we are experiencing internet connection problems. Also, the lack of compute or internet infrastructure in the Philippines. So that's why we are sometimes struggling with internet connection speeds, and yeah, and of course the internet connection is really essential to running the Bugseq's platform. And so might I ask, how in your side can this internet problem be addressed? So like in terms of running the platform, and
Yeah, that's a good one, because there's currently a lot of disparity or inequality in sequencing in the world. If you look at, for example, SARS-CoV-2 and where most of the sequencing data was generated, to date, it's been in high-income countries predominantly. And so closing that gap to make sure that actually we're sequencing equitably across the world is really, really important. And we believe in that strongly at Bugseq. And so, yes, Bugseq is a cloud-based internet platform. And so you do need to have internet access, but there are lots of measures that we're taking to improve reliability and accessibility to it. And so some of the things that are very simple, for example, genomic data can be big, but it can actually be compressed into small sizes. And so not only can you compress the files, which is pretty simple with command line tools or even user interface tools, but you can do things like remove unnecessary data before upload to the cloud, so that you strip away 90% of the useless data and keep the 10% useful data. So for example, for metagenomics tools, for metagenomics applications, we've open sourced a tool which removes all of the human sequencing data before upload to the cloud. So not only is this important to reduce the data size and the transfer, but it's also important for privacy concerns, because we know that there's lots of ethical challenges about uploading human sequencing data to the cloud. And so we avoid that altogether by stripping that data out before it ever leaves a lab that sequenced the sample. So those are just some of the things. There's a whole spectrum of other things that we're working on to enable broader accessibility of Bugseq. And again, I would say if you are having challenges with submitting data to Bugseq, I'd connect you with our tech team. We've even done things where like people can ship us the data on a hard drive if they need. Obviously, that takes a lot longer, but you can go back to all these kind of old school methods of transferring data and large volumes of data. Sometimes just shipping a hard drive or a thumb drive is the best way to get a large volume of data across the world. And so there's lots of options, and we want to work with you to enable that integration with systems.
Thank you very much, Sam. I can attest that Sam and their team at Bugseq is really supportive. As we've worked with them in the 𝗕𝗏𝗺 subgrant project. Actually, what we did to address the challenge of internet connection is that we are thankful to the 𝗕𝗏𝗺 grant, also funded by Bill and Melinda Gates Foundation. We are really thankful for, they have provisions for us to connect, like fix our internet connection infrastructure here at PGC Mindanao. So what we did was we hooked up our network to the UP Mindanao internet connection network. So, and also we are also thankful to UP Mindanao for that opportunity, and also to the 𝗕𝗏𝗺 subgrant for providing us with some funds to be able to do that. So yeah. So maybe two more questions. So actually I have one in mind, another question in mind. What was my question? So sorry, I forgot my question. So maybe let's have this one final question for Sam here. Actually, this was also asked in a previous okay, so I already remember my question. So actually I talked to you about this already. Have you seen like is sequencing and assembling of eukaryotic genomes becoming more common because of course, compared to prokaryotic or bacterial or viral genomes, eukaryotic genomes are really much larger. And so I would like to ask, is sequencing and assembling, or basically like biosurveillance of eukaryotic pathogens, becoming more common nowadays? And how can you or we address the challenge of assembling a much larger eukaryotic genome? So do you have any thoughts on that?
Yeah, so we're pretty fortunate that a lot of the advances in human genomics are spilling over into microbial eukaryotes as well. So a lot of the advances in assemblers or sequencers can be transferred to microbial applications. And so there are several key eukaryotic pathogens which are kind of on a lot of people's radar right now. The big one that we see at Bugseq would be Candida auris. And so this is a fungal species. It's a yeast that causes mainly hospital-acquired infections, and it can spread on things like blood pressure cuffs, stethoscopes, dirty hands, and it's very resistant to antifungal drugs. And so many of the common antifungals that are used to treat patients don't work against Candida auris because they'll be resistant. And so tracking the spread of this pathogen between patients in a hospital setting is very important to control its spread. And so that's one of the pathogens that we're working on analysis at Bugseq. And there are different approaches. For example, you can align the reads to a reference genome to look for mutations. You can do de novo assembly as well. And so especially on like some of the newer sequencing platforms like HiFi, like PacBio HiFi, where you can actually resolve chromosomes because the sequencing quality is so good and so long. But you can even do similar things, not kind of in the same way, but on Illumina or Nanopore. And so I do think that the future is bright. There's still a lot of work to go. It's less developed on eukaryotic pathogens than it is on prokaryotic pathogens.
All right. So yeah, so that sounds promising. Yeah, because we have like something going on, Sam, at Bugseq, and yeah, so that's really promising to hear. So maybe the last question. This has been asked in a previous lecture series. So how do you envision the future of public health or applied microbiology bioinformatics?
Yeah, so I think the future, and we think the future is going to be where the sample preparation and sequencing is fully automated. And so this really frees up people to work on the stimulating things. And so instead of, all the pipetting, we can get a robot to do that. We can do that at much higher volumes and much more cost effectively. And then we can sequence every sample that comes in the door of a public health lab or a clinical microbiology lab. And so whether that's direct from specimen, looking for that, tuberculosis, which is buried in a sputum sample, or whether that's from bacterial cultures, so it's been grown at another lab and then they send you the bacterial isolate. There's a future coming where we're going to sequence everything. And there's a lot of information to be had from sequencing. And so that will inform the bioinformatics, where again, we can really enable that future where an appropriate tailored analysis, which can actually impact clinical and public health care, will be almost basically automated from sample all the way through to result. But that's not to say that that's going to replace bioinformaticians. There's going to be an even bigger role for bioinformaticians in the future, where this is automating a lot of the kind of like regular routine stuff, but the number of non-regular, non-routine things continues to grow as well as we sequence more. And so there's always those interesting cases, outbreaks that need tailored investigation, really like a diverse array of questions where the bioinformatician is going to be the key, almost quarterback to answer those questions. So that's how we see the future at Bugseq.
Wow, thank you for that, glimpse of the future for bioinformatics. So as much as we want to, me personally, I have actually still a lot of questions to ask you, Sam, but unfortunately, we have a limited time right now, so for this talk. So thank you very much, Sam, for this has like become like a consultation meeting of sorts as well. So we have and thank you for our participants for your questions as well. And to wrap up things, so I'd like to take this over to JP. So thank you.
Thank you, Sir Paul. What a meaningful discussion, Doc Sam and Sir Paul. Thank you for that very engaging and interesting discourse. As much as we would like to, we do not have enough time to answer all of your questions, or if you still have other questions in mind that was not asked in this session, you may contact our speaker or through the PGC Mindanao email address so that we can relay your question to the speaker. At this point, we would like to give our appreciation to our moderator and speaker. So I would like to request Doc Sam and Sir Paul to please turn on their cameras. On behalf of our program director, we'd also like to request our bioinformatics technical expert, Doc 𝗕𝗵𝗃, to turn on her camera as well. All right, so in this awarding of certificate, let me read the citation. Philippine Genome Center Mindanao, College of Science and Mathematics, University of the Philippines Mindanao, awards the Certificate of Appreciation to Sam Chorlton for being the resource speaker on bioinformatics in applied microbiology, the last remaining barrier, during the Bioinformatics Lecture Series held on November 28, 2022. Signed, Dr. 𝗕𝗿𝗃, Technical Expert, Bioinformatics, PGC Mindanao, and Dr. 𝗕𝗿𝗁 Program Director of PGC Mindanao. Let us give a warm hand of applause to Dr. Sam Chorlton.
Thank you. And I just want to thank the PGC Mindanao team as well. It's been an absolute pleasure to work with them, and thank you for hosting this webinar.
It's a pleasure,
Thank you so
Dr.
much,
Sam.
Sam.
All right. Also, we would like to give the certificate of appreciation to Mr. Paul Lorenzo Gaite for being the moderator during the open forum of the talk on bioinformatics and applied microbiology, the last remaining warrior by Mr. Sam Chorlton during the Bioinformatics Lecture Series held on November 28, 2022. Signed, Dr. 𝗕𝗿𝗃, Technical Expert of the Bioinformatics Division, PGC Mindanao, and Dr. 𝗕𝗿𝗁, Program Director of PGC Mindanao. Thank you so much, Sir Paul.
Thank you, PGC Mindanao, for the opportunity. It's been a pleasure.
Salamat, Paul.
Thank you.
Once again, thank you to the both of you. At this point, I would like to request that you leave your cameras open for a photo opportunity. So please ready yourself and show us your best smile. One of our staff will give us a cue for the picture taking.
One, two, three, smile. All right.
Thank you, Doc Sam. Ma'am Chimi and Sir Paul, we'd also like to request the rest of the Bioinformatics Division staff to open your cameras for a photo opportunity. Again, one of our staff will give a cue for the picture taking. All right. All right.
1, 2, 3, smile. All right.
Thank you so much for that. At this juncture, we want to give our participants exciting prizes for joining our 2022 Bioinformatics Lecture Series. With that, we have 2 questions, and the first one to get the correct answer by typing the response in a Zoom chat box will win the prize. Can I get a thumbs up reaction from everyone? Are you all ready for our Q&A? We will be giving you exciting prizes. All right, I see a number of thumbs up already. So our first question, the winner will win an exclusive on-site laboratory tour at our institution. So the question is, how many lecture sessions did we have for this year's Bioinformatics Lecture Series? Again, how many lecture sessions did we have for this year's Bioinformatics Lecture Series? Wow, our viewers and participants are very quick in typing their answers. So I think all answers are in. And the correct answer is 4. We have 4 lecture sessions occurred every Monday of the month of October. So congratulations to 𝗕𝗿𝗃𝗃𝗃. You have won an exclusive on-site tour at PGC Mindanao. One of our staff will contact you for further details about your prize. Congratulations! Next, we have our second and last question. The winner will obtain a limited edition souvenir from PGC Mindanao. Again, the winner will obtain a limited edition souvenir from PGC Mindanao. And our second and last question is, how many years is PGC Mindanao been operating since its inception? Again, how many years has PGC Mindanao has been operating since its inception? All right, I already see a number of answers. All right. So all answers are in, and the correct answer is 3 years. PGC Mindanao started in the year 2019. So the one who got the correct answer first was Abdul Majid Dagung. Congratulations to Abdul Majid Dagung. You have won a limited edition PGC Mindanao merchandise. One of our staff will contact you for further details. Congratulations once again to our winners, and thank you so much to our audience for actively participating in our mini game. Now, we'd like to hear from our participants on how we did in today's activity. On the chat box, our staff will be providing you with the link of the evaluation form. Please fill them out before the end of the day. All respondents will receive their certificate of attendance once the survey is submitted and evaluated. In the event that you do not receive your certificates, please email us at pgc.upmin@up.edu.ph. For future events and to know about our services, please visit the PGC Mindanao website or our various social media accounts. You may also contact us through the number shown or email us through pgc.upmin@up.edu.ph. Our activity today is the 4th and last lecture of the exciting 2022 Bioinformatics Lecture Series. Thank you all for tuning in from the first lecture last November 7, the second lecture last November 14, the third lecture dated November 21, and up until today, our last lecture session. Watch out for more of our activities, and we hope that you continue to patronize PGC Mindanao and join us in the future. Again, on behalf of the Bioinformatics Division, I'd like to thank you all for attending the 2022 Bioinformatics Lecture Series of Philippine Genome Center, Mindanao. I am 𝗕𝗶𝗍 Lumabao. Have a wonderful day ahead of you and stay safe, everybody. See you all soon. Goodbye.